sick, so please don't sneeze on each other as much as you can. In fact, we are going to have some of the presentations are going to be pre-recorded, and I'll be reading to the best of my ability some of the slides for you. This has been a very difficult start of the semester. I'd like to spend a few words about Ross Clark, who departed very recently. Thank you, Diana, for this really cool photo. I haven't seen it before. Ross Clark, as you know, passed away on August 5th, and we're still recovering from this really sudden and terrible experience. And Ross was a student here in Mosellange. He graduated in 1996 in Mike Foster's lab. And he worked for about 10 years as an environmental scientist at the California Coastal Commission, and then in 2010 he returned here to establish the CCWG, the Coastal Central Coast Wetland Group, and he has been an incredibly successful colleague and friend and inspiration for all of us, students, faculty. So Sarah Stoner, CCWG, wrote something really beautiful on their Instagram and Facebook page that I'd like to read for you. She was very kind to sharing this with me. Ross wasn't just a leader, he was a heart and soul. When he founded CCWG, his vision was to advance wetland science and management along the Central Coast. Yet, Ross built something far more profound, a close-knit family committed to improving our environment. Ross didn't just create a work family, he ensured family in every sense came first. He treated our families like his own, always making sure we knew they were as important to him as the work we did together. When his daughter were younger, he would leave the office in the middle of the day to be there for them, whether it was to pick them up from school or to make smoothie when they weren't feeling well. At that time, I didn't fully grasp the importance of those moments, but now, as a parent myself, I really appreciate the example Ross set: family first, work second. Ross was a man of vision and boundless energy, with the habit of never saying no. His calendar was often triple booked, much to our frustration, but he still managed to be there for everyone who needed him. Ross was the life of the party, with a gift for connecting with people. His vision was always clear and inspiring, even if the finer details weren't exactly his specialty. But that was part of his charm. To Ross, who gave us so much of himself, we love you, we honor you, and we'll do our best to carry your vision forward with the same dedication you showed every day. We extend our deepest gratitude to the many friends, colleagues, and members of our extended CCWG family who have reached out during this difficult time. Your support means the world to us. So that was Sarah and the CCWG team. And I'd like to finish this introduction reminding everyone that if you want to contribute to our Family Fund, you can scan this QR code, or you can go on the GoFundMe page if you can get a picture of that, or I can send you a link. But actually, Kevin, I think recently sent, no, Kevin has, so I'm not. Okay, you sent a link to the whole community.
Yeah.
So, yeah. So, thank you, Ross, for everything. We'll miss you, but, you know, here we are. So let's do some science together, okay? Okay, so for some of you, this is the first time. This is our in-house open house with fast talks. It's a very intense, 66 minutes, circus of long activities. I don't know how else to call it. You're gonna have an overview of everything that's happening here. It's always fascinating to learn how much is going on here at Mosellange. Mosellange plus CSUMB faculty. So actually the way you see the series of presenters delineated here is that the faculties are mixed. So we are 20 people actually from both faculties. A really good, large faculty group to mentor our graduate students, and then we have a group of researchers as well. So 2 minutes presentation, I'm be timing it, and no questions. And please be next in line, be ready for the next one, so we're gonna get this this through in a little longer than 1 hour. And 30 seconds, I'll wave at you. At 10 seconds, you're going to see me walking towards you. At 0 seconds, I'll grab, actually the mic is over there. So you can keep talking about just getting your next person to overlap. Okay? So, and since I guess I'm yellow, I'll start first. Okay. And I'm going to set the timer. Oh, it's 11 minutes. That's not right. There we go. Okay, well, so again, welcome to the Inner House Open House. My name is Ivan.
Your time's up.
That's a star, all the way down there. Okay, I want my 10 seconds back. So, my lab is the George Oceanography Lab, and those are the lab members in my lab. My students, Rachel Clifford, who actually defended last Friday, and Lena Johnson, Mason Jones, Marcelo Pilix, Heidi Vasquez, and Kayla Vidal, that's co-mentored with Amanda Khan. And also I have a lab technician, GIS specialist, Charlie Andrews, works here in Alcasur. And as several of you know, I'm a kind of a geek on instrumentation. That's what we do a lot in my lab. I have a series of instruments that use either lasers or X-ray instruments to measure, see, it goes fast. Right. To measure composition and mineralogy of anything that's marine, mostly dead stuff. And our motto, as you know, is gotta leave the rocks. So if you look at the upper right corner, you see what this is all about. And I didn't know there were animations. So those are some of the projects we're currently doing here in my lab. So one of them, the main one, is actually looking at the transformation of biosilica. So we're looking at the carbon pump deep in the marine sediments. And especially we're looking at the biosilica that comes from biogenic activity are turned into rocks. And that has a huge implication for the way our planet actually works in terms of plate tectonics. And also we have local projects, two main ones. One is to map the head of the Maury Canyon. You see some really cool maps by my student Marcel Pilix that will be defending pretty soon, as well as marsh restoration in Alcasur. And this is my time. So next in line is Liz.
Thank you. Hi everyone, good to see you again. I think I saw a lot of you here last week. Great to see you. So I'm Liz Alter. I am in my now 5th year at CSUMB in the biology and chemistry department. And I am an evolutionary biologist. Our lab works on all manner of evolutionary and ecological genetics, and we have sort of 3 themes to our lab. The one that is most relevant for all of us here at Moss Landing is the evolution of species in the genus Sebaceae, so the rockfishes. We have a project that's sort of a holistic look at diversification in rockfishes, phylogenetics, speciation genomics, and morphological diversification. I have 2 students here, Parker and Jordan, that are both working on that rockfish project. We also work on freshwater fishes. You'll see we do a lot of work in the Congo as well. We do some work on marine mammal and other marine megafauna using ancient DNA. And we also have a program in environmental DNA. Most pertinently, we are working on a project funded through UCOP, UC, Office of the President, for climate resilience around the Pajaro River Valley and using eDNA to map biodiversity in that area. And I also teach molecular ecology and evolution at CSUMB. Molecular ecology is a class that if you're interested in population genetics or evolutionary genetics, encourage all the grad students out there to enroll. Thank you.
All right.
Hi, I'm Lydia Baker. I just got here this month, so I don't have a lab yet. I was working at University of Miami for 3 years and worked up a lot of my programs there. I specialize in bacterial interactions with marine animals. sharks. We're getting really far in that project. But I have a good long history of working with master's students to complete projects in an incredibly short amount of time at the University of Miami's Accelerated Master's Program. We do it in 3 months. You do the classwork for a year, you do your project in 3 months. So yeah, I also have worked a lot with bioluminescent bacteria, so I have ongoing projects with a collaborator at Cornell working on the evolution of bacterials and ions in anglerfish. But I also work in evolution of viruses and looking at viral genes as well. So basically, if you want to learn about DNA, as long as it's not eukaryotic, I'm the person to do it. If you have any questions about any of that work too, I'll talk your ear off for like an hour and a half.
Sorry, Baker with K, not with CK. Just change it last name. Okay, so there's another new faculty member from CSUMB who cannot be present here, Garrick. He sent me 2 slides and a recording.
In terms of some of my projects that I've been working on, a lot of these are looking at kind of cascading interactions. I've done work in benthic coral reef systems, looking at how epibiotic worms and amphipods alter the shapes of the corals. They change their morphology, and the indirect effects of that on things like coral predators and coral-associated fish. I have kind of a long-going project now looking at coastal gulls and their feeding interactions, both in their tidal and in the kelp canopy. I have a couple students now starting a project looking at how bird abundance in Elkhorn Slough affects rates of trematode infections in mud snails, and particularly how the presence of piscivorous birds seems to be related to infection rates in snails. And then I'm also just starting a new project, starting to survey the biodiversity of epivionts on intertidal mollusks. So we've looked at them on snails and chitons and mussels, trying to get a grasp of how prevalent these interactions are and the diversity of organisms that basically use living mollusk shells as a substrate. I look forward to working and meeting you all sometime soon. I'll be giving a seminar in the near future, so hopefully I'll be able to meet you all in person and talk a little bit about my research. Thank you.
Look forward to meeting you wherever you are. Tom Connolly unfortunately cannot be present today, so he asked me to just show you the slides and kind of go through the different points. As you know, he's a physical oceanographer here, and he works on currents and ocean waves. And it's actually very interdisciplinary, so he works a lot with students that approach different aspects of oceanography. He likes to go out at sea, like most of us, and to use oceanographic instrumentation, and also interpretation of remote sensing data. And so those are some of the projects he's working on. As you know, he's directing the SANCUS here in the Coastal Ocean Observation in Monterey. And he also works offshore San Luis Obispo, Alcoslu, and his students are working both locally and along the California margin. I'm sure if you want to know more, you can contact him directly or take his class, which is actually, I think, physical next semester. This semester, I'm totally lost. I don't know anything on the class. Next semester, okay, oceanography is next semester. See. Yeah. Okay, so Luke Gardner is our next speaker.
Hi everyone, I work on aquaculture. I have a research faculty appointment here, and I take students and I teach classes. I teach aquaculture as well as co-teaching marine resources and policy management. And my other title is Sea Grant Extension Specialist. So if you guys have any questions about Sea Grant, feel free to talk to me. We work on a bunch of different projects here in the lab. We work on basically trying to grow things. So it doesn't matter. We work on finfish, shellfish, seaweeds and things like that. But yeah, we have a presence in the aquarium room. We grow a lot of microalgae in there to feed various filter feeders. So we work on purple hinge rock scallops, trying to develop that as an emerging species for aquaculture, also looking at purple urchin ranching. Do some work on abalone, IMTA, fish aquaculture in the past. We work on sablefish. even doing work on rainbow trout and different feed formulations, monkey-faced brickleback. And then of course we do a fair bit of restoration aquaculture here as well. So we work with Elkhorn Slough to raise Olympia oysters, to put them back out in the slough to restore that population, and also with Bodega Marine Labs and nymphs on Whitehall and, like I mentioned, alternative feed work. And then we've got a few different techs and students in the lab. Andrew, Devin, they're techs, same with Alex, and then students Kirsten, Noah, Jacob, and Luke. Yeah, if you have any questions about Sea Grant or aquaculture, feel free to ask.
We can do this. Okay, Mike cannot be here, and he recorded a message some time ago that asked me to play for you guys. Oh, hold on, Mike. Go back, sorry, because it opens the page in my web browser, then I have to drag the web browser here, then I have to click here.
2 minutes and 40 seconds, you're going to cut it off.
The only way to do that is
Hi everyone, sorry to miss all of you today. It's Mike Graham. Unfortunately, I'm out of town doing field work in San Luis Obispo County, so I can't be there for in-house open house. I want to say hello to everyone, give you a little bit of a heads up of what's going on. We're still doing our aquaculture work and seaweed work in the phycology lab. Welcome all of you new students. My class this year is going to be taught in spring. I'm currently teaching a marine botany class, which is 100% online for undergraduates. But I'll be back teaching my grad class in the springtime. Lots of stuff in our lab, a lot of students. Matt Elliott just graduated. We're really happy that it happened in the summertime. Shelby Penn, Sloan Lofi, JC Lanza, and Mina Satari are hot on his heels. They should all be finishing up in the next few months or so. Jesse Carrizo and Bennett Bugbee are still in the lab doing kelp work. And we should be seeing a defense from Bennett coming up very, very soon. Jasmine Niger is still doing her work on sea grapes. Roxy Garibay is working on giant kelp reproduction disturbance. Matt Hess is looking at the flow of animal pee through the holdfast kelps. Marina Hernandez is out there working on the ulva work with drones and on the mudflats. Katie Duncan is out looking at the bacterial responses of some bioremediation in the Elkhorn Slough. Jess Franks is working on a super cool seaweed that no one ever works on, called Stephanocystis, and so happy she's doing a lot of field work right now. Maddie McKay is still working on bull kelp with drone surveys. Chase Ebner is right in the middle of his thesis doing some abalone IMTA. Lucy Gaw is about to start up doing some oyster IMTA. And then we got our new students, Natalie, Bella, and Elika, who are all just coming in and getting all excited. So I'm really happy to have a vibrant lab. Andrew Kim is our postdoc that we share with everybody, and that's just what's going on with the beer pigs. I'm not going to be around very much during this semester because I'll be doing field work and a lot of remote work, especially doing this asynchronous class. But I want to wish everyone a fantastic semester. Hope to see you guys around when I'm in. And I just wanted to leave these last few moments of my time remembering one of the best Moss Landing students ever, one of the best spirits we've ever had, and souls roaming those halls, and my absolute best friend in the world, Ross Clark.
Mike.
Bye everyone, sorry to miss all of you.
In real life.
Hi everyone, Max Grant, Chemical Oceanography. In my lab we mainly work on trace metals and nutrient cycling in the marine environment, and our current goal is to move analytical chemical oceanography to a new era using microfluidics. And we're doing that because most of the techniques that are currently in use in oceanography were developed more than 60 years ago, and as such they require also highly trained analysts and are completely unsuitable for remote deployments or even unattended monitoring on ships. So we're trying to get away from that using microfluidics in my lab. That's a challenge we're trying to tackle. And we're using microfluidics because you can make instruments in a very small compact package, and you can actually use very small volumes of reagents and samples to do your assays back to your instrument. And that's why you see that drop here. That's roughly the amount of reagents we're using to measure one sample. We've developed recently a new instrument that we call Fiona, which we've configured for autonomous monitoring of silicate and phosphate, and we've deployed it in various places, including in the Southern Ocean. Here you see a nice silicate transect from south of Tasmania, pretty much showing you the position of the fronts of the Antarctic circumpolar current quite nicely in here. And also we use the Mars Landing Shore Station as a test bed to deploy all of our instruments as well. And in the meantime, we're still quite keen to get some autonomous shipboard analyzers for aluminium, which is a great tracer of terrestrial impacts into the oceans, as well as iron, because it's the most limiting micronutrient in the ocean as well right now. In terms of my lab, this is it. Right now, we're a pretty small group, but a mighty group nonetheless. We have Seamus that joined us in the fall of 2022 that really helped us out with the silicate analyzer we've developed, and now he moved on to carbonate chemistry and is working on pH. We have Carol Chen join us in the fall as well. She's our iron lady. She's working on an iron sensor and applying it to study the iron limitation mosaic in the Monterey Bay. Lauren Bain just joined us, and we'll figure out what she does fairly soon. And we also have a real chemist, Katie Bergman from SGSU. She's a grad student there, and she's working in collaboration with CCWG. Thank you.
Thank you.
Hey everyone, Scott Hamilton, Ichthyology Lab. We study fish. They're half of the vertebrates. They are the best by far. We do lots of things, studying from individual species interactions up to ecosystem processes, climate change and kelp forests and other systems, the effects of fishing and marine protection, and aquaculture. I've gotten wrapped into many aquaculture projects thanks to Mike and Luke. So some current projects that we're doing, studying the effects of ocean acidification and hypoxia on reproduction in rockfish. That's with Cheryl Logan. A lot of work that's aiding fisheries, so collecting data from ground fishes that are used to enhance stock assessments on fish life history and demographies. We're involved with some marine protected area monitoring, both in the surf zone and in the collaborative fisheries project which Rick started, and I've been helping run for the past couple of years. And then in aquaculture, we've been doing a lot lately, studying with bull kelp restoration up in Northern California, integrating multi-trophic aquaculture with abalone and seaweeds, and then as Luke mentioned, trying to farm monkey-faced eels. There's a lot of people involved, so it's not just me. I also helped co-sponsor numerous students with faculty at CSUMB, SJSU, and research faculty here at Moss Landing. So I have about 10 students in my lab. There's about 20 others that I'm involved with. So it's like a quarter or a third of the students somehow fish. Well, the new students who are just starting out, you too can join the cult of the rockfish. This is last year's induction ceremony. Those are actually dead fish on their heads. So, cult is not just for your MAGA family members anymore. So some of the people in my lab, here's the brains of the operation. So Nate Stimble's a postdoc in my lab. Kevin, Andrew, and Jake are technicians. Jake's also a student with Rick. Here's our eel farmer, Luke. Right, these students, Bruno, Dylan, Sam, Quinn, and Grace, like to torture fish and do the climate change studies. Here's Jonah. He's doing a bunch of age and growth work with rockfish. And then I have two new students in the lab, Caroline and Riley, and they're figuring it out, but they have a lot of great ideas. And now I'd like to introduce my cousin. Come on up.
Okay, first I updated my slides, but I did it too late, so it's not my mom's fault, it's my fault. But these are not my current lab members. I am Scott's second cousin. We didn't know each other that much growing up, but we've become very close in marine biology. My current lab members, I have two, Randy Barton, who's up there, who is finishing soon. She's doing some work on population genetics of marine parasites. And then two second-year students, Haley and Olivia, who are working on an eDNA project and Pismo clams. And then new student in my lab, Ailish, who's there in the back, who's going to do some work with me to see who's eating juvenile urchins. So the projects, I'm a marine ecologist. Generally, I use a lot of different tools. I have a background in population genetics as well, but I'm not a geneticist. I just play one occasionally and use it as a tool to answer ecological questions. I just got back from doing a Fulbright in Mexico for the last 6 months. So I do a lot of work in Baja California and a lot of local field work here. I also, as a CSUMB faculty, I work with a lot of undergraduate students, and I really enjoy helping graduate students at Moss Landing find undergraduates to work with. As a assistants and mentees and things like that. So if you're looking for potential undergraduates to work with, I'm always happy to chat with you about finding students. And our main projects right now are, intertidal community assemblages, looking at changes in abalone, urchin, and sea star populations, and also looking at recovery of pismo clams in sandy beaches. Happy to chat with anybody about ecology or organisms or anything.
Okay, so we have another CSUMB faculty member, Sal Jorgensen, who cannot be here today. He recorded a, oops, okay. All right, here you go, Sal.
Morning, everyone. Sorry I couldn't be there today. I'm teaching this afternoon. But I wanted to introduce my lab. My name is Salvador Jorgensen. I'm an assistant professor at CSUMB. I'm a marine ecologist by training, and my lab focuses on ocean predator ecology. We mainly study elasmobranchs and look at different population dynamics, develop tagging technologies for biologging, and look at interactions of predators and prey species in a community ecology context. So I teach a capstone class that is open to graduate students. It's called Ocean Instrumentation, in which we develop different tagging projects and build tagging applications. So this is a great place to come over if you're a grad student, check out what we're doing, find some interested undergrads that might be interested in helping out. I'm always looking for collaborations with the faculty members. So thank you very much.
He's quiet by himself. This thing is not working. Okay. Nate is no. Is Nate coming? He's not coming. Okay. So, next time Nate is another colleague of CSUMB, and does anyone want to say what he's up to in his lab, or next time? Next time. Thank you. Guys playing some sort of I know it's not an instrument. I thought it was like a tambourine or something. Amanda is also not coming today. She cannot be here, but she pre-recorded a presentation as well. And
as a whole, as a group, the lab studies three major focuses.
I don't know why.
As a whole, as a group, the lab studies three major focuses.
I know what's happening. Sorry about that. I think the first slide advances, the second slide is she talks. Let's try that one more time.
My turn. As a whole, three major focuses.
Thank you for
All right, I brought show and tell. I am Katie Leach. I am the librarian here, and I have some awesome new student assistants. Well, sorry, I have Ava and Heidi who have been working for me, and Elise who is brand new, to help me with all things library stuff. So the library is a standalone specialized library, so we do all the back end things that nobody sees and all the front end things, helping you with your research. I brought show and tell, grad student success collection, new bound theses. Our bindery closed during COVID and it took forever to get a new one, so yay, those will be on the shelf soon. Games, super fun. No? sabbatical working on georeferencing all those species. 55 plus years of MLM master's thesis, so that'll give us a new way of looking at the species. It'll be really fun. I'm excited about it. And then I just put up little information about the 2 libraries that we have access to here, so that is another thing that we can help with here in the library. So helping you navigate that 2 library situation. And that's all.
Hello, my name is James Lindholm. I'm in my 17th year at CSUMB. I'm the chair of marine science and the DAC program. Is this my second slide or my first slide? I don't know what's going on. Losing time. Okay. I work on a lot of imagery-based analysis, mostly looking at fishes as well as some selected inverts and how they're interacting with things on the seafloor. We use visual tools ranging from scuba all the way to subs to do landscape ecology. We're doing MPA monitoring, fun global project that we started when I was a postdoc, looking at coral reef fish interactions all around the world, how they forage collaboratively. And the newest thing that we started right before COVID set in was using VR. Recent graduate student Cameron Strickland defended his thesis comparing VR data collection to a visual survey by a diver, showed the metrics are comparable. So we've been exploring VR, but when COVID shut us down, we started working on a curriculum so the students at CSUB had something to do remotely. So we swam the VR transect through the kelp forest and the students extracted data. Now, for those of you, anyone's interested in broader impacts, we have a high school curriculum that we're launching, and we have student ambassadors go with us to launch the VR curriculum, and we're just working with local hospitals to bring VR to patients in hospice, chemo, dialysis, and palliative care, which is more rewarding than any of the science policy interface I've ever done. We have 3 students right now, 2 which came in during the COVID period and working with Scott and Amanda. Carrie is just starting this semester, and we're supporting and working with Star Lab significantly and with Jake Todd and Jackie Mohay. We have funding from state, federal, and private sources, all of which the majority of it goes towards supporting student support, and there's always room for more people who are interested, even if you're working in somebody else's lab. If you want to do some of this broader impact stuff, we'd love to see you on our end. Thank you.
All right, hi everyone. I'm Cheryl Logan. I'm a professor in the Department of Marine Science at CSUMB as well. I've been there for 12 years, not as long as James. I am a marine environmental physiologist, and I currently have 7 students, with Daylin up there at the top. Ari, who just graduated, no longer a student. I've got 2 new students, Emma and Matthias there. My 2 second-year students are Ava and Madison, and then Katrina and Nick are 4th-year students. So we are interested in the effects of climate change on marine species and marine populations, specifically looking at how different species are affected by changes in temperature, hypoxia, and ocean acidification. So we're interested in the mechanistic basis of stress tolerance, what physiological thresholds different species have, and how flexible those thresholds might be in the face of a changing climate. We also use that information and integrate it into models to forecast how species might be affected in the future using the output from global climate models. And we hope that this information can help to inform adaptive management decisions. So here are 3 examples of some of the projects we're working on right now. The first one is understanding mechanisms of heat and cold tolerance in corals in the eastern tropical Pacific that experience really wide ranges in temperature. These corals are extremely hardy, and we're trying to understand why and how and what the applications of those mechanisms might be to corals elsewhere in the world. I have Katrina and Ava and potentially Emma who are going to be working on that project. As Scott mentioned earlier, we've done a lot of work looking at the effects of climate change on rockfish, various life history stages. The current work is really focused on rockfish reproduction and the possibility of transgenerational plasticity in terms of whether exposure of the parental generation influences and helps the offspring or not, and if so, what those mechanisms might be. And the last project I won't talk about today.
That's you. Thank you, Martin. You're Martin. There you go.
Oh wow, you don't know that.
Yeah, it's
I
very
don't know
fast.
the fact, but
Yeah, me
anyway.
too.
So hi everybody, I'm Martin. I'm a postdoc in the Vert Lab. So I'm speaking on behalf of Birgitta McDonald. So in the Vert Lab, what we're doing is like mostly studying the physiology and the behavior and ecology of the vertebrates, mostly pinnipeds and big birds. And so, for example, we're like studying the diving energetics for the physiology or the diving behavior and trying to mix all this information to bring knowledge about their diving behavior. We have some projects that focus on the conservation too. So, for example, here too, project based on the pinnipeds to see how anthropic disturbances can change their behavioral activities. And to give more insight about what we are actually doing, so here are the project of the different master students we have in the Vert Lab. So Amber and Kaylee are working on the multiple stressor and tagging impact on the elephant seals. We have some students working on the arbor seals in Oslo here. Sierra is working on the sea otter behavioral responses to anthropic disturbances too. Vicky, Taylor and myself, we are working on the habitat use and the foraging ecology of emperor penguins, all using different approaches. And finally, Sebastian is working on the foraging strategies of the elephant seal using an isotopic approach. Thank you.
Yeah. Yeah, it's you.
I'm here.
Yeah.
I'm in.
Yeah, please,
Okay.
please.
My name is Sherry Palacios. I'm a faculty member with CSUMB, assistant professor there. I'm also a biological oceanographer, and I'm an alumni of Moss Landing Marine Labs. So it's nice to see you. That's right, I am. So these are some of my undergraduate researchers. I want to mention I have two thesis students here, Christian Martinez and Maddie McKay, and I'm happy to serve on committees as well. My research focus, let's see, oh, where's Sal? Okay, let's see. My research focus is on developing algorithms to apply to imagery to understand what's in the water and how is it changing. I was at NASA before I came to CSUMB. I developed methods using satellite remote sensing to look at the water in a bigger picture. I'm a biological oceanographer. Some of my questions here are deriving algal biodiversity from multispectral and high spectral resolution imagery, estimating nutritional quality of the phytoplankton for their grazers, or this concept of food from space, modeling seagrass productivity, because you can't get away from Elkhorn Slough for still 57 seconds, and optical modeling of plastic in the marine environment. If you're interested in talking to me about these projects, I'd be happy to. I also teach a class that will be offered this spring. It's called Biological Oceanography Projects. It's a remote sensing-based class, and it has a graduate level. And I welcome you to enroll in that class. I'd love to see more people from here working on that. Mike Wood works on a lot of questions related to physical oceanography and computational oceanography. My focus is more on biological oceanography and understanding remote sensing data. And I'll probably be using a lot of the PACE data, which is a new sensor that's gone up just in February. I'm also the director of the research experience for undergraduates at CSUMB, and many of the faculty here are mentors to our interns, but they leverage their graduate students to help to mentor those students. So if you think you might be interested in a scaffolded approach to learning how to mentor, please talk to me. Thank you.
Right.
Okay,
No slides.
no slide for me, technical difficulties, but I think we can do it. So I am Sarah Smith, the biological oceanographer here at Moss Landing Marine Labs. I've been here for 3 years now, starting my 4th year, and the lab is coming together really nicely. I have 3 students in the lab. Hannah McGrath, who's co-advised with Holly Bowers, actually primarily in Holly Bowers' lab. Dominic Kyle Epel, who is working on Elkhorn Slough and doing a really nice metabarcoding study of the phytoplankton and other protists that live there. And then I have a new student, Elise, starting this year, who's going to be working on a recently
funded project on Rise of Selenia diatoms, and that has a long cruise, 35-day cruise in blue water diving. So it's real, kind of uses a lot of the capacity that we have here at Moss Landing. I'm really excited about it. I do a lot of work with phytoplankton ecology and evolution using genomes and genome-enabled tools. So I teach the molecular biology class here in the fall. Some of you are enrolled in it, which is awesome. And then I teach biological oceanography in the spring. And I think that's it. I think I did it in 1 minute. All right.
Hi, my name is Diana Steller. I'm sorry I missed the new student orientation for some of you. I run the dive program here, but I'm also on the research and teaching faculty. So we have a great group here that supports the diving on, in, and under the water. And they are grad students, Jess Franks and Logan Early, who's helping run the San Francisco State Dive Program. We do training, we help with equipment, we support the diving operations here, and all of your scientific inquiries, and encourage you to not check your email, but go outside. I help teach classes, MS 105 in the fall. I also co-teach or have co-taught subtitled ecology, a periodic field methods course in Baja, and help with a lot of the collaborations between the institutions around Monterey Bay, which are rich in marine science, but also diving. I have worked with various students over the years, currently Sloan Lofley, Celine Dion, and Jess Franks on their thesis. And my research interests are in macroalgal ecology, kelp forest ecology, coral and algal ecology, and really expanding the access for diving for research to all of you. And so what I often say the first day is, I encourage you to do exactly what you're doing here, which is come in person and go up to people, and I'm talking most of the students, but also the faculty, meeting people and asking for opportunities to go outside. And when there are field trips that Rick Starr is going on, and with his group, go ask them. And so I really encourage you, there are so many opportunities at Moss Landing, but also at CSUMB and all around Monterey Bay with all the different research institutions. And so ask for opportunities. Be curious. And then we're doing blue water diving that Sarah just brought up. So in the next 3 years with her NSF funded research, we're going to be doing a training program in research blue water diving. So come join us.
Okay. Where's the arrow? Hi, I'm LML. I'm Mike Wood. Impersonated here by Ivano, or whoever is reading this. Hopefully they are putting on their best Mike Wood impression. My accent is going to be good. I'm one of the faculty members at the lab, and sorry I couldn't be there with you in person today. I'm in San Jose this afternoon because I have also a faculty position in the Department of Computer Science at San Jose State University. I teach a hybrid computer science courses, and Tuesday happened to be my in-person class day of the semester. So I'm at MLML. I'm in the process of starting the computational oceanography lab at the end of the hall, in front of my lab. If you see me tinkering or banging around in there, feel free to pop in. The last space has come a long way in the last year. We'll even have our own software soon. There are 3 students contributing to the project in my lab, including 2 from computer science and 1 from the physical oceanography lab at Moss Landing Lab. So it's basically, I don't know what the slides are showing, so I'll go to the next one. My personal research interests are related to glaciers and sea level rise, particularly in Greenland. I use numerical models and remote sensing heavily in my research, as well as in seabed observation when I can get to the Arctic. Through the computational oceanography lab, aka the cool group, I got it. I hope to bring expertise in numerical modeling and remote sensing to the lab. This semester I'm teaching a remote sensing class, and I'm in the process of designing a modeling class to add to the curriculum for 2025. If you have any interest in these topics or need help with code, don't hesitate to get in touch. I look forward to working with many of you. Bye. So We go now to the research.
Hello, I'm Scott Hess, Moss Landing Laboratories affiliate, research affiliate, and alumni of Moss Landing Labs. Welcome to Moss Landing Labs. I lead the U.S. West Coast leatherback turtle research and monitoring effort. Our core responsibilities to inform and the recovery plan for leatherback turtles include distribution and abundance through aerial surveys, collect genetic information for population structure, and perform telemetry to look at movements of leatherbacks within the California current and outside of the California current. And we also do some foraging, ecology, and health assessment work. As I mentioned, leatherback turtles are critically endangered, one of NOAA's species in the spotlight most at risk of extirpation within the next few decades. It's an extreme transboundary animal that originates from beaches in the western Pacific and moves across the Pacific to forage on the west coast of the United States. We monitor abundance trends, habitat use, and foraging behavior, and this information is used to inform domestic and international stakeholders, including the Pacific Fisheries Management Council, the California Department of Fish and Wildlife, and international bodies charged with conserving leatherback turtles. Our data is also used and contributed to dynamic ocean management tools and models. I'm located at Norte. We have opportunities for students to join in our aerial surveys. And once again, I welcome you to Moss Landing Labs.
Hi everyone, my name is Holly Bowers. I am stationed down in Norte, just like Scott. I'm his neighbor. And we are the Environmental Biotechnology Lab, and our main focus is harmful algae and phytoplankton in general. So without phytoplankton, none of this other stuff exists, right? Because we are at the base. So I have a couple of students who do their work in my lab. Hannah McGrath is working on 2 harmful algal species of Pseudonychia. And the thing is, they look alike under a microscope, so you can't distinguish them. And so you have to use genetics. And so she is combining these 2 species and possibly some others to look at competition. And so not a lot of studies have been done on that because you can't just look under a microscope and distinguish them. So she'll be diving in with some genetic tools to be able to look at who's winning out. And Olivia Pollock, she is working on looking at the effects of microplastics on both harmful algal species as well as just good old phytoplankton that we need out there. And so she's cutting up lots of plastic and sticking it into bottles with bugs and doing lots of different measurements to see how they react in terms of growth or potential toxicity. And so some of the projects that overlap with their work, and some I'm working on independently. At the top, we have a NOAA OTT grant now to take this new type of imager out and get some images of phytoplankton. And so the gold standard is sort of a more expensive instrument that gets put out. And so this is sort of the low cost version. And so I've got some recent pictures on the left there from a bloom of first Dolichospermum in Pinto Lake, just over in Watsonville, and that switched over to Microcystis, which you can see there are those blobby things on the bottom. I just rotated off being co-chair of the National HAB Committee, and we have a big report that just came out, so if you want to know anything about HABs, come see me. And we're also working with CCWG, EPA-funded grant to look at ag water growth. Thank you.
Hey everybody, I'm Dustin Carroll. I'm a PI here at Moss Landing Marine Labs, and I'm an affiliate scientist at NASA's Jet Propulsion Laboratory, which is down in Pasadena, California. My background's in physical oceanography and numerical ocean modeling, and I'm a Moss Landing alumni as well. So I've been back here almost 6 years now as a PI, which has been great. I have 2 main research tracks. One is I'm the lead developer for NASA's flagship ocean biogeochemistry model, which is called EchoDarwin. So I do a lot of model development, working with various NASA groups and doing science with that. And my other research focus is doing ocean ice and carbon interactions in polar regions, specifically the Arctic and Greenland in the last number of years, So I have field campaigns almost every summer doing work in Greenland. The next one will be next spring. We'll be going in the winter and drilling through sea ice in the winter to measure ocean properties and fjords. So right now I have 8 NASA projects and 3 NSF projects, so lots of work. The group is myself, 2 postdocs and an intern at NASA JPL, and then a variety of graduate students and colleagues. And my primary collaborators are at Scripps and MIT. And another really cool direction we've been going the last couple of years is I've been starting to use the NASA modeling technology to do marine carbon dioxide removal, in particular simulating it. There's so many companies out there right now that are starting field trials for this. They need a quantitative background and model to be able to do that, so they're starting to use Echo Darwin. So we have new NASA funding that's coming in as we speak, directly from NASA, so no proposal solicited, to start a program doing modeling of marine carbon dioxide removal. And just to show you, those are some of the companies that we've been working with in the last year that are all interested in doing simulations of marine carbon dioxide removal. Thank you.
Hey everybody, I'm Kevin O'Connor. I'm the new director of the Central Coast Wetlands Group down at Moss Landing here. We're down at the Sandholm Center, down at the bottom of the hill in the little barn down there next to the 2 little farmhouses. Our group, we have several full-time staff, Sarah, Jenny, Brooke, and Jess. We also work with Steve Cunningham, he runs the Nutrient Lab here, and we've got some other partners, Ariok and Stephen, who help us do restoration. 2 students assistants right now that we're working with, both me and Katie. And then we have watershed stewards, and we were a placement site for the watershed stewards program of the California Conservation Corps. So we have 2 stewards coming in October, Petra and Jaden. Our mission is to coordinate the advancement of wetland science on the Central Coast. And we do an assortment of work, wetland riparian monitoring and mapping, wetland dune habitat restoration. All these dunes that you see out here, it's one of our restoration projects, removing all of the invasive ice plant and restoring the native plant community. We also do a lot of estuary monitoring. We run the Estuary Marine Protected Area Monitoring Program for California. Climate change adaptation planning here in the Monterey Bay, as well as up in Marin County. We do a lot of work with the local farming community, implementing treatment wetlands to help reduce nitrate runoff into our local estuaries, and then overall watershed planning in the area. Some of our current projects, as I said, we've got the Estuary Marine Protected Area Monitoring Program. It's a collaboration between us, SQUIRP, and CSU Long Beach, and then up north we have UC Davis and Sonoma State. We're all monitoring 5 to 7 estuaries in each region. We have the Moral Coho Slough Management Enhancement. We're doing extensive restoration projects out in Moral Coho Slough. We have a new project funded by Caltrans to look at sea level rise vulnerability of the Highway 1 along Bolinas Lagoon between the towns of Stinson Beach and the Bolinas Fly. And then we were recently awarded, as part of a large grant to the region from NOAA, a $71 million grant from NOAA for a coastal climate resilience project here in the entire Monterey Bay. And we're working on a monitoring plan for all the projects, a restoration project in the Old Salinas River Channel, and Monterey Bay Living Shoreline projects along the crescent of the Monterey Bay. So, any questions about all that? We're down in the farmhouse at Sandholm Center, and look forward to meeting you.
Oh, yeah. Huh? Is that you?
Nope.
Yeah, you gotta go back a couple.
Oh, sorry.
She got scared so first.
I got confused.
That's okay. Everybody.
Thank you.
Okay. My name is Erica Burton. I'm a research ecologist at the Monterey Bay National Marine Sanctuary, which is a marine protected area that was designated in 1992 and extends from Marin County, just north of the Golden Gate, down to Cambria in San Luis Obispo County. It's about the size of Connecticut when it's included with our Davidson Seamount. And our mission is to understand and protect the coastal ecosystem and the cultural resources of the sanctuary. And our research approach is to determine the scientific information gaps, and then develop collaborations with people like you to study the issues, and then we interpret it for our decision makers and the general public. We have a large management plan that gets updated every 10 to 15 years, and so we bite off little chunks. So right now, these are some of our priorities, including getting a better handle on microplastics in the system. So
So we're sampling from the rivers to the beaches to the ocean. We just completed a climate change vulnerability climate vulnerability assessment. It's getting formatted right now, and we'll soon be embarking on a climate adaptation plan, and that's been done with regional experts in the area. We're trying to get a better handle on the soundscape so we can minimize human impacts to whales and to minimize ship strikes. This month, we just completed an iconic kelp forest action plan with regional scientists and our Sanctuary Advisory Council to find out what the needs are for monitoring, restoration, and recovery of our kelp forest right now. And we're transitioning from paper static condition reports to report on the health of the sanctuary to a more web-based approach. So we can, by serving up real-time or near real-time data to understand the health of the sanctuary. And the ways that you guys can get involved immediately is we are doing microplastic samplings on a quarterly basis. Our next opportunity is September 3rd and 4th. It's a great day out on the beach. We can train you there. Every 2 to 3 years we get NOAA ship time, and we go out to Davidson Seamount to do marine mammal and seabird surveys, and we're always looking for observers. And we're also starting to partner more with Cal Coffee. And you are all welcome to join a Research Activity Panel meeting that happens quarterly with our regional scientists. The next meeting is September 13th. We may actually be meeting here in December to be determined. Those are virtual right now. And I urge you to look at our science needs so that you can apply your own research to conservation science issues in the sanctuary. Thank you.
Okay, Dave Ebert is one of our affiliated researchers not here today. He's usually around the world. In fact, he's currently traveling in Timor-Leste, in Indonesia, searching for lost sharks and avoiding crocodiles. Successful on both counts so far. The largest crocodile seen so far is 17 feet tall. Work with local communities to improve food security, training local fishery personnel in shark and ray identification, found 6 critically endangered Laos-North Grand species, including the river shark that was tagged and released. I guess the one in the picture. Project supported by the Save Our Seas Foundation. He also is a very prolific writer. He has a new book out. It's called Sharks, Rays, and Chimeras of the East Coast of North America, published in May. In preparation of Ghost Sharks of the World with former Moslem student Dr. Jenny Kemper. 12 publications so far in 2024, including a cover article in Science this past March 2024, and found 2 new ghost shark species. So that's Dave Ebert. Wes. Oh, Wes also gonna come.
Hello, ML community. Apologies for not being able to be there in person today. My name is Wes Heim, and I direct activities out of the Marine Pollution Studies Laboratory. We do projects in marine, estuary, and freshwater environments, looking at habitat health and contaminant cycling, sediments, water, and biota. I have a list here of current projects we're working on. This first one we're finishing up this summer. It's a coastal survey of the California coastline, looking at contaminants in fish. We're also finishing up our EPA National Rivers and Stream Assessment this summer. I want to highlight this eelgrass bed survey project down in San Diego. That one's ramping up now, and lots of opportunity for collaboration and employment. So opportunities in MPSL, we do direct employment for field laboratory and data handling. There's opportunity for thesis projects and support using our lab and instruments. Equipment's always available for student use down at MPSL. Our location is the northwest corner of the ML Norte facility. We have a large staff of people that are excited to help and collaborate in various ways. I'm going to show a couple pictures here of us in
cannot be here today, but she like just to show you the slide here. She focused on biological fishes, oceanography, and larval fish, krills, salps, multi-species, zooplankton assemblages. And so she's looking at these long-term data sets to understand, the effects of climate change on these marine species. And between 1986 and 2009, she was a contract scientist at NOAA, an Arctic Marine Living Resource Program. And especially in the past, she was very involved with the research and student mentoring here. And she's kind of slow burner right now, still affiliated to our lab, and but always very happy to work with students that are interested in working on any of those species. So if you are interested, I can put you in touch with her. She just wanted you to know, guys, that she's around. And and there are 2 slides. Let's see, collaboration, abundance variation, Carinaria japonica. Okay, next. Marco. Sorry,
I'm Marco Segala. I'm the other half of the Marine Pollution Studies Lab. We started in the early 1990s after Rusty Ferry graduated from Moss Landing. He wanted to stick around, so he started our group. Wes is half of the group. They started before us. Rusty came in a little bit later and kind of broke off and started his own. We provide scientific expertise and support services for environmental research and monitoring throughout California, mostly for the State Water Board, but also EPA and other groups, nonprofit as well as private industry. We have a much smaller group than Wes. I'm the project director, Stacy Swenson. She's our data management and quality assurance expert. Will Hagen is up in Seattle. He's our quality assurance guy as well. And then we have George and Michael Radojevic. They're our technical wizards, their father-son combo. I just hired Michael earlier this year to help us out with some of our data management projects. We do have students, both past and present. Justin Gill, Sam, Roxanne, and Ilaria are going to help us with the SAB project that Wes mentioned down in San Diego this fall. So we do have some experience or some needs for help. I think Justin sent an email out to the students. If you're a diver and you want to dive down to San Diego, there's opportunity for that. If you want to work in the lab, we just bought a leaf meter, so we're going to test that out. Next year we have a big EPA National Coastal Condition project where we'll be sampling all the bays and estuaries throughout California. We have our SAB project, like I mentioned. We'll be sending samples to the nutrients lab for sediment and tissue analysis. We also do freshwater bioassessments for Sierra Pacific Industries on all their timberlands to monitor the health as they're performing their operations. We do work in San Francisco Bay. We do data management. That's our primary focus right now. For the Water Board, we help Preservation Inc. with the ag order on the Central Coast. We also help with QA for the Delta Regional Monitoring Program. And we have a Central Coast Data Navigator that helps get out all the data for the Central Coast if you're interested.
Okay, so Rick cannot be here today. Oh, Nate. I don't know. I have no idea.
You can go first.
Oh, I got the alphabet.
Okay, cool. Thanks for sticking around. I'm Nate. I'm a postdoc in Scott's ichthyology lab. I work on several projects. I've really enjoyed my first year here at Moss Landing, working with a number of scientists and graduate students and undergraduates. I was primarily hired to work on fisheries, and that's one of the 3 core themes I work on, including ecophysiology, fisheries, and global change biology. I am currently analyzing data from the California Collaborative Fisheries Research Project, CCFRP, to look at dynamics in inshore fisheries. In particular, I'm interested in looking at spatiotemporal patterns of various fisheries metrics like biomass per unit effort or fish maturity as a function of distance to nearest NPA borders. And so basically trying to generate more insights into how things like habitat characteristics, MPA characteristics, and fish life history traits and spatial structure interactively determine fisheries outcomes. And I'm doing similar work, kind of moonlighting, working for Cal Poly on offshore fisheries, using a completely different sampling methodology that I'm excited to see is yielding very similar patterns. And I'm addressing kind of three core questions. First, how do fishers respond spatially to MPA closures? In other words, do we see evidence more of fishing the line behavior or ideal free distribution? How does the cost of transit and habitat influence fishing distribution? And finally, how are those modulated by constant versus dynamic closures? This summer I had a really good time mentoring an REA student through the CSUMB program that Sharia Palacios mentioned earlier. I had the privilege of working with Julia Zapadka on a cool project utilizing the tent at the Shore Lab to look at multi-scale impacts of upwelling on purple sea urchins. I'm also working on an ecophysiology and conservation aquaculture effort involved in the sunflower sea star, which is an endangered mesopredator. And I mentored a student involved in the URISE program, which is funded by the NIH. So I invite and welcome and hope to interact more with the community here. So thanks.
One and only.
Thank you. Hi, I'm Rick Starr, and my group in the Fisheries and Conservation Biology Lab work on the distribution, relative abundance, species habitat associations, and movements of fishes, primarily those that are commercially and recreationally caught. We gather information on these species, we develop new tools, we developed a new BOSS video lander to go down to 400 meters deep, working with Magari. We worked with James Lindholm's group to create some new video landers for sampling areas in and outside of MPAs in the nearshore area. And we work on larger conservation issues as well. We're heavily involved in the development, the design, and implementation of the MPAs in California. So I have two students who are hopefully going to finish it within the next 6 months or so. Jake Todd, who is, as Scott said, is leading up the CCFRP field work this summer. It's a great opportunity to get out and see what fishes are like in the nearshore areas and in MPAs. Jackie Mohe is working for a contracting firm, a consulting firm in Santa Cruz, and she is working on some of our BOSS video data for her thesis, and hopefully it will be done. I also put up Kate Ressender up here, one of James's students, because we've been working closely with his group on these development of these video landers to use in nearshore areas. And Kate, Jake, and I are headed for Portugal next week, where we're going to spend 3 weeks on a small boat, the Santa Maria Manuel, deploying video landers on the seamount, a couple hundred miles off the coast of Portugal. And that's my time.
This is a small boat. Almost there. So Alison Steeper is another one of our research faculty. She cannot be here today. And so she asked me to read a few words for you. So sound travels well underwater, which makes it important for communication, navigation, and foraging for marine species. We study cetaceans' behavioral ecology, including sound production of individual animals, as well as their underwater movement and behavior, and how it is associated with sounds produced and received. We do this using suction cup pads that record sound and sometimes video, in addition to body movement. We also study the effects of anthropogenic sound of marine species, including general characterization of marine soundscapes using passive acoustic recorders. Right now, Alison has one student, Jack Barkowski, who used passive acoustic monitoring data to describe presence and movements of humpback whales on the US West Coast. She encourages new students to get in touch if they're interested in discussing how biological tags or passive acoustic could help inform their study question. So, Alison's going to be around for any questions. And now the final group, Claudia is presenting for Jeffrey.
Oh yeah, it's up there. It's not here though. Okay, so I'm Claudia, I'm the wheat lab. It's just Jeff and me. Jeff can't be here today, so apologies. We have currently 3 big ongoing projects. And he just got back from a cruise to the Juan de Fuca cleft segment. Amanda Khan and 4 ML students tagged along on that expedition. We collected water samples near hydrothermal systems, and they're being analyzed. And these things have a way of repeating themselves, so that I'm pretty sure we're going back and there will be opportunities to tag along again. That's the first project, and then I'm going to get Oh. I know he was frozen there. Okay. And then we developed a new sampler that's basically a push core on steroids. It's out of titanium with 1 million little parts. It will be tested this fall by us, which means me. And I'll be standing out there with a drum and some clay and mud, and I'm trying to drive that thing in and sample the sediment. We're after the water, so thrice once you get it out normally. And that will happen next year, or 26, with the Oregon State University coring group. And again, there will be opportunities for people to come along, and we are open to the exact location, so whatever fits in your studies, who knows, maybe we can collaborate on something. And then a third project that's an ongoing thing, we are deploying our Osmos samplers, which take continuous water samples without any power supply by utilizing osmotic pressure. And they're deployed on moorings in the Gulf of Alaska. We have done that before. One time we got samples back, or got our samplers back, and I have them in the lab. And the first one, unfortunately, the whole mooring got ripped out by a ship or something and vanished. The hope was to recover them and the mooring, but no, it's gone. But that's gonna keep going, and yeah, that's about it. That's all I got for you.
Well, that's it. Thank you for coming. We'll see you again on Tuesdays, 4 p.m. for seminar series. We have a pre-house lineup. So please come to our seminars in person as much as possible. We want to be here together.